Okay, hi everybody. I hope that we are all ready to start. So welcome to the webinar on RNA-seq data analysis. I'm Eija Korpelainen from CSC.
And I'm Maria Lehtivaara from CSC. And today we are indeed talking about RNA-seq data analysis using Chipster software. This is our very first Chipster webinar, and we are really keeping the topic very brief today. Oops. Just a moment, some technical difficulties here. Thank you. Okay. Yeah, indeed we are keeping the topic very brief, and we are focusing on the main steps of differential expression analysis of RNA-seq data. Usually our courses of this particular topic are like 1 to 2 days, so indeed this is going to be a short introduction kind of presentation. We are hoping to get some feedback from you guys. I already posted the feedback link to the chat window, so feel free to fill that up. And if you have any ideas of possible topics that we could be presenting in the future, please feel to share those, because we would really like to use this opportunity to teach more detailed stuff also about RNA-seq to you guys. Okay, but yeah, today we are briefly introduction Chipster software, and then talking about the differential expression analysis of RNA-seq data. And Eija will now shortly present Chipster software.
So, like some of you might know already, so Chipster is a free software developed at CSC, and it gives access to hundreds of analysis tools. So we don't invent the analysis tools ourselves, but we just take ready-made tools and put them behind this easier to use interface. What is important to realize is that Chipster is a client-server software, meaning that when you use it, you will have a little client program sitting on your computer. But when you run analysis tasks, they are actually running on the server side. So all the heavy computing is happening on the server, which now in our case is here at CSC in Espoo. This is what the client program looks like, but we will look into that in more detail soon in the live demonstration. And this slide here is just to remind you that, well, today we talk about RNA-seq data analysis, but actually you can also analyze many other kinds of data using Chipster. Then a couple of words about how you import data to Chipster. So the data can come from your own computer. It can be individual files, or you can import a whole folder. You can also, this is especially handy with sequencing data because the files can be very big. So if the sequencing core facility puts the files behind a URL on the net somewhere, then you can actually download them directly from there to the server. So there is no need to pull them down to your own computer first, but they can just go directly to Chipster server. So you would, in Chipster, you would select this tool in the utilities category. And of course, Chipster also allows you to open not only files but full analysis sessions, so you can share sessions with your colleague. Then another important thing to remember with sequencing data is that it's really quite okay to give compressed files, so zipped files. So there's absolutely no need to uncompress them. It's better to keep them compressed because they are big already, so they travel faster when they are zipped. And Chipster analysis tools can happily use them zipped, it's not a problem. The other point I wanted to make here is that today we go through the whole workflow. So we start from the raw reads in fastq format, and then we align and have count files. But if the core facility, for example, already gives you BAM files, it's quite all right to start with those in Chipster, and also if you have count files, that's fine too. So you can jump in at different points of the analysis. And obviously it's important to save the work that you have done. In Chipster you save it in analysis session, which contains the result files, but also extra information about the result files. And you have two options for saving them. So you can either save them locally, which means you are saving them on your computer, or you can save them in the cloud, so on the server side. You can do both, but just remember that when you are saving in the cloud, we are not going to store them there forever. So it's always a good idea in the end of your analysis to also make a local copy. So far we haven't deleted anything yet, but that day might come when things get really full. And finally, when you are doing analysis, sometimes you get an error message. So something is wrong, the analysis tool is not running properly. So then it offers you a possibility to contact support. It will ask you, do you want to contact support? And if you say yes, you will get this kind of window here, where you can type a little message to us, put your email, and then importantly, tick this box here. So when you tick this box, which says attach data and workflow information, this allows us to open your session so that we see it exactly the same way as you see it. So we can run some tests with your data to solve your problem. For example, we can see if one of your files is corrupted or whatever. So rather contact us this way than send a separate email, because the good thing with this is two things. So first of all, you can give access to your session, because otherwise we cannot access your session. And also it will automatically copy the error message, so you don't need to copy that. And now Maria will continue from here, moving to the actual analysis.
Yes, so indeed that was the short description of Chipster, and now we are moving to the actual data analysis part. So this is the really simplified overall analysis steps. Okay, there seems to be one question in the chat box. I will answer to that after this part. So okay, so here in this image on the top, you can see the gray reads. So that's your raw data that you get from your sequencing facility or your sequencing device. So it's small parts of your original RNA. The first thing that you have to do, well, of course, you can do some pre-processing and some quality control there. But first, actual step that you do, you align those reads to the reference genome. So the black bar here represents the reference genome, and these black bars are now your reads that you're aligning to your genome. Next, you match these aligned positions to known genome positions. So for example, here we have gene A and gene B, and we know now that these reads here belong to gene A and these are from gene B. And the third step in this workflow is to count how many reads each gene has. So for example, now gene A has 6 reads and gene B has, well, 11 reads. And from these results, you get these columns from this table. So these are the read counts per sample. And the final step, when you have done this workflow to all your samples, so this is now one sample, and then you would have several for different groups. Then you combine those read counts into one big read count table. And then you can move on to comparing the actual sample groups. So for example, here you have 3 control samples and 3, well, some other samples that you are comparing to the control group. And then you do the statistical differential expression analysis. So you actually compare the counts that you just prepared. So for example, here in gene D, you can see that nothing is really changing. The counts are very low, so there's nothing interesting going on there. No difference between the groups. But for example, with gene A here, we can see that okay, there's more reads in the sample group than in the control group. So then that's a upregulated gene. So these are now the really main steps. So alignment, matching alignment positions with known gene positions, counting the genes, the reads per gene, and then the differential expression analysis. Now let's add the actual analysis steps and tools and the file formats that we need. So now this coloring matches to those that we use in Chipster, so it's maybe easy for you to follow later on what we are doing in Chipster. So we are doing quality control in several steps. So first we do quality control for the raw reads using the fastq tool, and then later on in the alignment step we do some quality control also for the alignment, and then later on for the whole experiment. In the very first phase, these raw reads are in fastq files, so you get those from your sequencing facility or sequencing device, and you usually get as many fastqs as there are samples, so there are a bunch of these files. Then you can, if you want to, you can do some pre-processing. For example, in our example session we use Trim-O-Matic to do some trimming to our reads. After that you still have a bunch of fastq files. Then the next step is the alignment, for which in our example session we use TopHat tool. And after alignment, your fastq files are transformed into BAM files. So now you have a bunch of BAM files instead. Then we do the quality control with RCQC to get some information on how the alignment went. And then we move on to quantitation. So now we are counting the reads, and for that we use HTSeq. And from that step we get a bunch of read count files, which are just tables basically showing this first column here for each of the samples. The orange step here is combining the read count files into one larger read count table, as seen here. And for that we use, in Chipster we use this define NGS experiment tool. And then we do again quality control using PCA or clustering to see how the whole experiment went and are we happy with our results at that point. And the last step is the differential expression analysis, where we use here DSeq2, and from that we get a list of differentially expressed genes. Okay, so now let's start to look a bit more into detail of these different steps, and maybe go through some common issues in those particular tools. So first we have this quality control step, and we are dealing with fastq files. This is briefly what the fastq files look like. Luckily you don't have to look at them too much, but just to show you, there's always 4 lines per each read, and they look like this. So there's twice the read name, and then the read sequence here, and then this weirdly looking quality coding for all the different bases in your read. We will be looking more details to that later on, so don't worry too much. If you want to know more about this format, Wikipedia has actually quite a good page regarding that. And like Eija already mentioned, you remember that you don't have to unzip your fastq files. Chipster can deal with zipped files just fine, so you don't really have to check this file in detail. And if you're wondering about the qualities, like maybe you want to check those, well, you can use the fastq tool, which was the first tool in our list, to see how your reads look like. So this is just one example of the images that the fastq produces. So here you can see the quality values in nice numbers and in this sort of traffic light kind of setup, so you don't really have to worry about those ASCII characters at all. And then next step is alignment, and Eija will talk a little bit about that.
And perhaps what we forgot to say in the beginning is that we are also recording this webinar, so I can see in the chat window that somebody has trouble hearing us, so at least in any case then he or she will be able to listen to the recording later. But anyhow, let's move on now to the next analysis step. So the next step now is that we want to match the reads to the reference genome to figure out where they are coming from. And in our example today, we are doing with a TopHat aligner. So there are several aligners, but for RNA-seq data, you have to select one which is able to cope with splicing, so in order to cope with introns. So in my little drawing here, I have, this is like a part of genome here. We have exon here, and an intron in between. So some RNA-seq reads come from within an exon, but some go across exon boundaries, like the purple one here. So the aligner has to be able to place this correctly in the genome. And then there is one thing that you need to remember. So there are actually different lab protocols to prepare reads, and some of them are stranded, some are unstranded. Maria will talk more about this later, but basically the stranded protocol is able to keep the information whether your read comes from the actual mRNA strand or the other strand. And if your data was produced with a stranded protocol, you need to know which one so that you can select the proper parameters at this point. And Maria has actually made a very nice manual page which explains this in more detail. Then there is another thing that you have to be aware of. So, so sometimes, well, depending on what sort of sequencing machine was used to produce your data, you could have either just one or two fastq files per sample. So if you have single end, you have one file. If you have paired end data, you have two files. But then if your data was produced with this NextSeq machine, then it is very likely that you will actually have several fastq files per one sample. And then what you want to do is you really want to align all of them in one go. So in one TopHat run, you want to keep all of them. And now then it depends. If your data is single-end data, then this is super easy. You just select all those fastq files that belong to that one sample and run TopHat, and no questions asked. If you have paired end data and you have several pairs, then what you have to do is you have to prepare this file name list files first. So I have explained here the steps what you have to do, and I also have a picture here. So here I have 3 pairs. So for paired end data you have the 1 files and the 2 files. So this stuff here is a pair. So what you would do is you would prepare a name list for the 1 files and a name list for the 2 files. So here I show what that looks like. So just a very simple file name list, and there is a tool for that, and more explanations in the manual. Once you have done the alignment, out comes a BAM file, which looks like this. So at the top there is a header listing like all the chromosomes that were in the reference genome, and then after that come the actual alignment information for each read. And so all this would be in one line. So 1 line per each alignment. And note that there can be more alignments that there were reads that went in to the tool, because one read can align to several places in the genome.
Okay, then moving on to the quantitation step. So we are now counting the reads, and in Chipster we use HTSeq for that. There are a couple of things that you should know. So for example, multi-mapping reads, so those reads that are mapping to several locations in the reference genome, and ambiguous reads, they are not counted when using HTSeq, so you don't have to worry about those, but it's good to know that this is what happens. Also, again, it's good to keep in mind that if you're using this stranded protocol, like Eija already mentioned, keep in mind that you select the right counting mode. And again, we are linking to the manual page for this, but I'm explaining it a bit more detail in a few slides. So, okay, I was talking about these ambiguous and multi-mapping reads. So on top here, we can see the ambiguous situation. So here we have the reference genome, and we have a gene A here on the other strand and gene B on the other one. And now if we have a read here and we don't know from which strand the read originally came from, we really don't know from which gene it is originally from. And thus, well, this situation is called ambiguous, and we cannot decide, and we are thus not counting this read to either of these two genes. If we, however, have the stranded or directional library preparation, the information of where the read originally came from, so was it from this strand or from this strand, that is available. So we know that, okay, if we know that it was from this strand, then it possibly can't be from this gene B. So we can exclude that from the analysis, and we know now that it's definitely from gene A. So using the stranded library prep helps you with these ambiguous reads. But you have to remember to choose the right parameter for that one. The multi-mapping option, which I was talking about, well, that happens too. So your reads, unfortunately, can map to multiple locations in the genome, and then again, they are not counted. Okay, still trying to explain the stranded or directional RNA-seq sample prep. So yeah, there are several protocols available that do this stranded option. So for example, TrueSeq, stranded, NEP, ultra-directional, Agilent SureSelect, strand-specific. These are all directional kits. So as you can see, it's usually visible in the name that, yeah, indeed they are directional or stranded. And if you're using some of these, make sure that you choose the right parameters. It's important to remember both in the alignment step and in the quantification, so in TopHat step and in the HTSeq step. Unfortunately, the parameter naming in these different tools is a bit incoherent, to say the least. So, well, if we go through the TopHat options, well, the unstrand parameter is pretty clear. So then you don't know from which strand your reads are from. But then if you have the directional, if you are using one of these kits, the option is either this one or this one, so the first strand or second strand. And now it gets a bit complex. So for example, if you're using TrueSeq stranded or NEP ultra-directional, you are using this DUTP method to maintain the strand information. And in that case, it goes so that always the first read, so if you have paired end data, the first read is from the opposite strand, and the second read is from the original RNA strand. And if it's the other way around, then, well, the first read is from the actual original RNA strand, and the second read is from the opposing strand. If you have single end data, then it's always only the first one. So in this case, it's from the opposite, and in this case, it's from the original RNA strand. Now, here's the same parameters for HTSeq. So if you have this option in TopHat, you should use this option in HTSeq. So for first strand, it's stranded reverse, and for second strand, it's stranded yes. And yeah, well, for unstrand, it's stranded no. So this makes sense, but this is kind of, you just have to know. We are trying to improve our manuals to make this more clear, and indeed we now have the special manual page explaining this issue in more detail if this was a bit complex to understand at this point. Okay, then Eija will talk about combining individual count files.
Okay, so when the HTSeq tool has been run, as an end result, you have a pile of these files. So for each sample, you will have one file where it lists the number of reads matching to each of the genes. So the next step then is that you want to combine all these individual files into one table like this. And for that, there is a tool called, in the utilities category, called Define NGS Experiment. So this tool creates not only this table, but also another file, which is called phenodata file, which is actually a very special file and has a big importance in Chipster. So phenodata file is the place in Chipster where you describe your experimental setup. So otherwise, Chipster has no idea which samples are control samples, which are treatment samples, or whatever. So this is where it happens. So it's essentially just a table. It's created automatically. It has certain columns by default, but you can add more. And so there are some super important columns, and especially the group column here. So that is there by default, and if you are going to do differential expression analysis, you absolutely have to fill in this column. So what you fill in there is using numbers, you describe your experimental groups. So rather than, for example, writing control and cancer, you use numbers so that the smaller number is for the control group and the bigger number or numbers, if you have several groups, are then for the different treatment groups. And so here, for example, I have control samples marked with 1, DPN treated samples marked with 2. Of course, if you have a more complex experiment with, say, time points or pairing, like I have here, so different time points, again marked with numbers, different patients, again marked with numbers. Then the other column you might be interested in is the description column, because here you can write text, whatever you want, so that it's the text that you want to appear in the visualizations. So when you will make plots of this data, like for publications, how do you want to call the samples then? And then I pass to Marin.
So this is the third of the quality control steps that we had. So we had one quality control for the raw reads and one for the alignment, and this is the third. So this is the experimental level quality control. And we do this to check whether the experimental groups separate from each other, whether there's some batch effect visible there, or if some of our samples is an outlier. And there are several methods to do this, for example, principal component analysis as seen in the picture here, and then there's clustering. And we will check these out also when we go to the Chipster soon. Then regarding the statistical testing for differential expression, so sort of the last step in our workflow, there are a couple of things to take into account. Well, first of all, biological replicates are really important. You cannot really do your analysis properly if you don't have enough replicates, so please have at least 3 per group, so 3 for the controls and 3 for the samples and so on. Yeah, and then raw counts are needed to assess the measurement precision, so please always use the raw counts for when doing the differential expression analysis, and do not use the FPKMs, for example. Then there's the multiple testing problem as usual when we are testing thousands of things at the same time. So in our case, genes. So as you can see here in the DSeq2 result table, we have several columns, but the most important ones are the fold change column here and the adjusted p-value column here. So please do note that you should use always this p-adjusted column and not the p-value column when you are sorting your genes. So our DSeq2 result table is actually already sorted based on the p-adjusted. So the most differentially expressed gene is on the top and so on. And the log fold change, well, that's simple as it is. So it's the fold change. So if it has a minus sign in front of it, it's downregulated, and plus then it's upregulated. If you want to separate them for different groups, for example. A few words about the visualization.
Okay, so before we move on to Chipster soon, I just wanted to point out that you can also visualize your reads in the Genome browser. in Chipster. So what we have here is, so we are in chromosome 18, which is showing here. We are in this position, and here we have annotation from the unsampled database for this position. So we have there a gene, SYT4, which has several transcripts. Now below here we have tracks from our samples. So these are the alignment files, the BAM files from several samples. They belong to two groups. There is one lymphoplastoid cell line here, and then an embryonic stem cell line here. And now you can see that, yes, so what we have here is read coverage. So Chipster is automatically calculating how many reads do we have in these different positions, and then it's drawing these peaks. So yes, we can say that definitely it looks like these embryonic stem cells do express this SYT4 gene, whereas the other cell line doesn't. And down here we have one result file from differential expression analysis, also showing that yes, this gene was considered as differential expressed.
Then still, before moving to the Chipster, maybe the most important thing from this webinar, so where to get more information and help if you need. Well, to get help, you can always just email us to chipster@csc.fi or send a support request like Eija described earlier. We also have several example sessions in Chipster, just like the one we are soon going to check out. So those are really useful as well. In the Chipster webpage, so chipster.csc.fi, we offer several manuals and tutorials, so you can check those. In YouTube, we have, well, this webinar will go there as well, I think, but we have several tutorial videos for different topics, and for example here we have RNA-seq playlists, and we are wishing to get some ideas from you guys as well, from the feedback of what kind of videos would be useful for you. Also here in the course material page in csc.fi/web/training/materials, we have this course material link there. So there's a RNA-seq data analysis for bioinformatics, RNA-seq with Chipster, and RNA-seq with command line tools and R if you want to maybe like do some exercise tasks or follow some tutorials. So that's the place to find those. And then we could move to Chipster and check this out there as well.
And yeah, I just quickly show this slide. We come back to this later, but basically there is also a list of Chipster tools that just to remind you what tools you run in what order. But let's go to Chipster first to see how things are done there. So so I'm in Chipster now, and I have one example session open. So this session is called Webinar 1 RNA-seq. And this is also available for you. You can open it later if you wanted to. It has two data sets here. There is one ENCODE data set, and then there is one Drosophila data set here. And I have explained what the data is and what we have done. But so in general, if you are not familiar with Chipster, so on this side here we have the files. So here I have all the files I have produced when doing analysis. The same files are showing here as boxes. And so, for example, if I select the BAM file, it will also highlight there. And as you can see, boxes have different colors. So colors refer to the analysis tool categories that are here. So the tools are, first of all, grouped in 3 sections. We are in the NGS section today. And then within the section, they are in categories, so that they would be easier to find. So let's run a tool, or in order to run a tool, Maria can run it later, but so you select the input file, and then you go to the correct tool category and to the tool, and you can either click run or fiddle with parameters and click run. And you can always find more help, so there is a little help here, but you can find more help from the manual by clicking here. And otherwise, so as you will notice, as I click through the files, this changes. So this tells details about that particular file that I have selected. So what tool created it, when, what parameters did I use to run it, and what visualization options I have for that file. And finally, I also wanted to point out, we talked about that support request, which you can send when you get an error, but you can actually send it even if you don't get an error. So if you just go to help, there is contact support, and if you click that, it's exactly the same window. So if you just want to ask us a question and say that, hey, can you look at my session? I have this problem, blah, blah, blah. I'm not quite sure what to do. You click this, write your message, your email, and click OK, and then it will come to our Chipster team. So nobody else will see it, just our team. So, and otherwise about the data in this session, so we actually have now 2 data sets. So up here in the ENCODE data set, we show the full workflow starting from the fastq files, the raw reads, ending in the differential, in the list of differential expressed genes annotated. But here we only have 2 replicates, which is, well, there are no replicates, there are only 2 samples, so that's a bad thing, and you should never do things that way. So in order to show you a proper session which still runs quickly, we have down here another data set where we have more samples starting from the count step. And finally, there is one lonely PDF file here. So this is actually the slides that we are showing now. So if you go to example sessions and select this one, you will also see the slides there. But so now Maria will talk about the quality control.
Yeah, okay. So starting from the first session up here, or from the first workflow up here. So first we indeed have the fastq files. And now the first thing that we wanted to do was to check the quality control, the quality of the raw reads. So indeed we check, select this fastq file here, and then we go to NGS quality control and read quality with fastqc. And if we check the parameters, well, there's not much there. It's just a fastq file type, which we select like this. And then we can click and run the tool, like so. You can see here that the tool is now running, and soon the result file should appear somewhere here. There we go. So now this is a HTML file, which we can open in our web browser, like so. And if you have ever seen a fastqc report, this is just like the ones you've probably seen. So we have the traffic lights here, and this is the same thing that we already saw in the slides, but it's now looking a bit not that nice. And all the other images are here as well. Then if we go back to Chipster, well, I can now delete this file if I want to, because I already have it there. I was just demonstrating. So that's how you delete a file, like so. So as you saw, the quality didn't look that nice, and that's why we decided to do some trimming for these particular reads. So we use this tool, Trim-O-Matic. It's under the pre-processing category here, and there you can see it. So we use this tool to trim our reads a bit, and as a result, we get a fastq file again, and it's now zipped, and we don't have to worry about it because it's okay with Chipster.
Yes, so this trimming business is really a matter of taste. Some people do it, some people don't. It's not absolutely necessary because, of course, if you don't trim the bad bits away, then the read will not align in the next step. So you will also get rid of it that way. So now moving on to the alignment. So if we go back to the list of tools, so now we are in this category here. And so for the alignment, we give fastq files. So we could give this one or this one. They are both fastq. And then we need to select the aligner. And as you see, there are lots of different aligners, but we want one of the TopHat ones. So there are also 4 TopHat aligners. So you have to select based on what kind of data you have. Is it single-end or paired-end? And also, is the organism that you are studying such that Chipster has reference genome for it? So if you are studying some super rare organism, we might not have the reference genome, but then you can upload your own. But so now in our case, we have single-end reads, and this is human data. So, so here is the reference genome list. And yeah, as you can see, we have quite a lot of reference genomes there. And otherwise, the important thing from here is the library type. So I can actually move this a bit closer here. So the library type, if your data was stranded, this is unstranded, so I don't need to change it. And so I'm not running TopHat now because that would take a while, even for this small data set. I still want to point out that if you have paired end reads, then you have slightly different parameters. So there is still the library type, the strandedness is important, but also it will ask this one, what is the expected distance between your reads? And this really depends then on the fragment length that was used in the sequencing lab, and also the read length. So this is something you have to set according to your data and ask the core facility what length fragments they have.
So there was a question.
Okay, so there was a question regarding the reference genomes. Ridul is asking if one can align also against the older version of the human genome. So that would be here. So they have slightly different names. But otherwise, if you had really something very exotic that we might have never heard of, then it wouldn't be in this list. And it might be also not a public genome if you are studying on something where the reference genome has not been released yet. So then you might want to use your own as well. All right, so now, so when you have run TopHat, then you get, so these red ones here are the result files from TopHat. So there is the BAM file, like we discussed already. So, so the header and then the alignment lanes for the reads. Then there is also another one which has extension BAI. This is an index file that is needed by some analysis tools, but otherwise you don't really need to look at it. You actually can't look at it. Then there is a PET file containing junction data. But then this particular one is important, TopHat summary file. This one tells you how the alignment went, so it's a good idea to look at this one. So it will tell you how many reads you put into the alignment. So this one was very small set I had here. Normally you would have like 30 million. So here I have like 86,000. And then it tells how many of those were mapped and how many of those that were mapped actually were mapping to several places in the genome. So this is an important one to check. So if your alignment percentage is super low, like I don't know, 6% or something, then clearly something went badly wrong, and you want to start checking different things.
Okay, then after the alignment comes the counting of reads. So we are using HTSeq, which is found here under RNA-seq category. Here on the top we have HTSeq, and there too we have quite many parameters that you should check. First of all, there's again the reference organism. Then there's this is quite important as well. So there's the chromosome names in the BAM file. Do they look like this? So is it just one or is it CHR1? In Chipster we have the default of just the number, but you want to check this as well, especially if you're bringing your own BAMs and not creating them in Chipster. Also, if you're using paired end data, this is the point where you have to remember to check that yes, you're using paired end. And then to the stranded option. So here we have the stranded parameter in HTSeq. And here we are explaining them. As you remember, they were the reverse, yes and no, but we are also explaining them in a bit more detailed manner to make it maybe easier. And we are also describing them here in the this parameter text. And if you go to the help page, you will see see the whole manual there as well. So resulting from this HTSeq, you get two files. You get the actual count for this one sample. So now we have only two samples, and this whole workflow is for the first one of them, this HTSeq sample. So the count looks like this. So there's the IDs, and then some information, and then the actual count. So now you see only zeros here, but if we organize this table according to the counts, you can see that, okay, indeed for this first ID we get 7,503 counts. So actually that many reads were mapping to that particular location or that particular gene. So then the other file that we get from HTSeq is this sort of summary or info file, which we can view as text. And there you can see a couple of numbers. So there are the not unique alignments, which I was describing in the slides. So those reads that are not aligning uniquely, so there are multi-mapping reads, and there are always some number of those. Then there's also the no feature number. So those reads that are not mapping to any gene in the reference genome. And now this is a clue for you. If you get a huge number here, you might want to check the stranded parameter. And this is also one point where you can test if you selected the right parameter just by running the HTSeq with the different parameters and checking with which option you get less no feature reads, and then you should maybe go with that one. There's also the number for the ambiguous reads there. Then Eija will describe on how to combine these count files.
So once you have completed this thing for the different files for the different samples, then you should have a TSV file containing counts for each sample. And next you want to combine them. So what you do is you select all of them, and you can select several files keeping the control key down. So now I selected the two. And then you go to the utilities category, and there pick the tool define NGS experiment. And here then you can tell various things. So for example, does your individual files contain genomic coordinates for the genes? In my case they do, so I say yes. And then the most important one is this count column. So what is that column called where the counts are? And in my case, because this data is from Chipster, it's called count. So I can run this one now. This one doesn't take long because I want to show you that when the result comes and when you get the phenodata, Chipster will remind you that you actually have to do something with the phenodata. So, and always when a job is running, you can check by clicking on this view jobs thing what is happening. So now it's telling me that then there is a wow, there is a problem with transferring the files. That's weird. Okay, well, never mind. So this is the kind of error message you get if something goes wrong. And there are two buttons. So one is show details. So it says that it took too long to be sent. I don't know, something else something really big might be happening at the same time somewhere else. And then it gives you this option contact support. So then we are back to this thing that I explained earlier. So this would be where you send us a message. So now let's not worry about that too much. What I would have liked to show you is that when this thing comes, so these two yellow files, in the phenodata file, you would have a little exclamation mark here saying that you have to do something about it. And when you open it, you would also have an exclamation mark in the group column saying that you have to fill it in, because this would be empty. So you have to tell what are the experimental groups. Now this, like I said in the beginning, this is a very silly example because it's only 2 samples, there are no replicates. So that's why I added this other data set here, because I wanted to show you what a real experiment would look like. So now we jump from this point to this point with another data set. So here all these things beforehand have happened, and now we start from this point. So let's have a look at the phenodata here. And we can see that we actually have 7 samples in this experiment. There are 4 untreated samples and 3 treated samples. This is actually a Drosophila experiment with a knockdown experiment of one particular splicing factor. And then there's other information here. So there is also a fact that some samples were sequenced single end and some paired end, and I have also indicated that with numbers in this column. And if you look at the actual count file, here you can again see the different samples. So here we have the gene names. These are flybase identifiers for Drosophila. And then how many counts does this gene have in the different samples that we do. So you can see that the count levels vary quite a lot between the genes. So now Maria will continue from that, discussing the PCA plot.
Yeah, so indeed, now that we have this proper set of samples, so 7 of them, we can run this quality control with PCA and heat map. So it's under quality control, and it's the bottommost option here. And as a result, we get this PDF file, which you can view in Chipster. So this is the PCA plot, similar to the one that you already saw in the slides. So indeed, you can see that here the blue ones are now the untreated samples and red ones are the treated. So you can see that, yep, they are separated nicely. So your treatment is indeed having some effect to the samples. But on the other hand, you can see that, okay, there's maybe something else going on as well, because they are separated with the second principal component as well. So there's something going on in this direction, and now actually we know that it's the way that the samples were sequenced. So this is one kind of information that you can get from the PCA. Also, if you wouldn't know that there's this batch effect behind, you would maybe suspect that this is an outlier. So that kind of thing you would also see here. Then if we move to the actual differential expression analysis, so we used here DSeq2. So again, if you go to the RNA-seq category, you can see here several options. So we use this DSeq2.1 now, and as a result, you get again a bunch of files. If we look at the TSV, so the DE list DSeq2 TSV, we can look at it as a spreadsheet, and you can see the actual read counts here in the first columns, and these are the identifiers. These are not now ENCODE identifiers, but they are the flybase ones. And here in the right, you see the actual statistical values. So here's the log fold change, and here's the adjusted p-value. And as you can see, this table is already sorted based on the p-value. So the most differentially expressed gene is on the top, and so on. And we used here, as you can see from here, so these are the parameters that you use to run the tool. So you can see that the cutoff for adjusted p-value was 0.05, or I'm not actually sure if you can see it, but it is. So that means that these are now the top genes, and here we can see that we have 700 29 rows in our data, so that's how many differentially expressed genes we now have. DSeq2 also produces this summary file, which we can view, and it's just showing you briefly, like a quick glimpse. You can see that there are 350 upregulated genes and 380 downregulated genes. You have some number of outliers and some number of low counts, and you can also check the total number of reads that were used as input. So,
okay, so we have pretty much completed this. Maybe one thing I forgot to say earlier is that so up here, so I didn't discuss these files here, but they were also differential expression analysis, but it's not very meaningful if you don't have replicates. So I don't want to look into them in a lot of detail, but I want to point out that so here in the result files, so you have these ensemble identifiers for the genes, which are a bit awkward, so you can always annotate them. So there is a tool in the utilities category, annotate ensemble identifiers, and when you run it, then what you get is for each ensemble identifier, you also get the gene symbol and a little description. So this is maybe something you might want to do to your result list. So maybe, so let me see, there is a question if we are going to upload the presentation.
yes, we are. So this whole
it's also here.
Yeah, so this whole webinar, so the video is uploaded in YouTube once I get it edited, so later on today, hopefully. And then this example session that we have been looking in Chipster, it's in the example sessions. Can you maybe show how you open an example session? So you go to File, Open Example Session, and then there should be quite a bunch of different example sessions, and this is the webinar one. So there you can open this exact session, and indeed there you have the PDF with these slides there. Also in the original event page, I have listed some course materials, so there you can see a bunch of those. Okay.
There are also other questions regarding exome-seq data. There is actually well, there are some tools in the variants category now. There will be more soon because the GATK tools are coming there. But perhaps we could run another webinar on that topic and just at this point maybe say that well, there are tools for exome-seq data as well. And maybe still I can leave this slide here as we are wrapping up. So just to remind you what we did today. These are the tools that we were running. So Maria was showing how to check the read quality with fastq that produced an HTML report. Then optionally you can trim the reads, you don't have to. If you have paired data and lots of fastq files per sample, you want to make those file name lists. We didn't need to do that today. But then the steps you have to do and the tools you have to run is TopHat, either for paired end or single end reads to produce the PAM. Then you can check quality at this point with a tool we were not showing today, but we could run maybe a separate tutorial on that. Then you count reads per genes with HTSeq, get the count file, combine the count files, and then do the PCA plot to have an overview of the experiment, and then do the actual differential expression analysis. And you can also annotate the gene lists in the end. So
there were a couple of more questions. If we can go back to Chipster. There was a question of how, can you show again how to get the differential expression gene list? So it's basically probably the last step. So if we choose the combined count file, like so, and then we go to the RNA-seq, and we used here now DSeq2, and then there are some parameters. And the most important thing here indeed is to remember to select the group column here. And if you have some additional experimental factor, like in our case, that would be the read type, we could select that one as well. Yep. And then the cutoff p-value, that can be now 0.01, and then we just run the tool. Well, we won't do it now because we have already run it. So then if you go to the TSV file, that is now the list of the differentially expressed genes. And if you check it as spreadsheet, you can also maximize it to see it more nicely. So this is the file that you are looking for. And if we go back, we can also, if you want to now give this to our colleagues, for example, that can be exported from Chipster. So if you just right click it, and there's this export option. So then you can later on open it in Excel if you want to study it more.
So with export, you export one particular file, but otherwise what you can do is you can actually save this session. So you can say save, either save local session, and then it will save it on your computer, or you can say save cloud session. But remember that the cloud sessions don't live forever, so at some point you also want to save a local session. So once you have saved the session, you can also give that to your colleague.
Then there's one last question regarding HTSeq data. So there's some old presentation in title, and indeed we have had some courses some time ago, and now the question is that are you going to have a video of HTSeq data using Chipster? That's a good idea.
Yes, we can definitely do that. And so, well, yes, so there's a HTSeq tool category here. And otherwise, there are also, meanwhile, if you want to get started, there are example sessions. So if we go back to example sessions, there is this one here. So this is NGS, HTSeq, Stat1, peaks, pathways, and motifs. So that goes from the BAM files to finding the peaks, and then also doing pathway analysis for the peaks and finding shared sequence motifs in the peaks. And we probably have somewhere also the course materials, so we could actually put it into that session.
Yeah, so I guess we are running out of time. So thank you so much for your questions and all the activity. And we would really love to get some feedback and also suggestions for upcoming webinars. We already got some, so thank you for those. But here's again the feedback link if you wish to fill it up for us.
Okay, so I guess that was it. Well, so thanks for listening, and I hope to meet you soon in another webinar or during a face-to-face course.
Thank you. See you.
Bye-bye.